change gears and I'll get ready to turn things over here to Home, where we can have a presentation on dynamics with Redcraft. So I'll just quickly flash up one slide here as a brief intro. So as you can see here, Home has his bachelor's from Michigan Tech University and his master's and PhD from Purdue with these degrees in electrical and computer engineering. Home then went on to do postdocs with Peter Albelsheim and James Prestegard, and he worked on structure determination of carbohydrates and computational methods of protein structure determination. Home is currently faculty at the University of South Carolina, where he's in the departments of computer science and engineering and biomedical engineering. He's the associate chair of research and also the associate infrastructure director in the area of visualization. And I think that kind of shines through in some of his software design and his careful thought into how he presents things. I've got a scale on our intro slide here just to kind of show, I think that Home does a really nice job of kind of balancing the computational and experimental components of his research. And today we're going to hear about Redcraft. So without further ado, Home, thank you so much for joining us again and for all the contributions you've made to the NMR Box community and for the instruction that you give us so generously. We really appreciate having you. Thank you. Home, you're muted, Home.
Okay, we should keep track of the score of how many times I'm going to screw that particular feature up. So, well, thank you, Adam, Jeff, and the entire NMR Box community for giving this opportunity. I'm very happy to present this Redcraft and structural calculation from NMR constraints. Is something that I do for fun, actually. It is part research, part just fun. So let me just make sure, I guess I should have done this first. I'll reach out to Adam. You can hear me clearly. Is there any disconnect?
No, everything looks good.
Okay.
You're not sharing screen at the moment, but we can see and hear you just fine.
Great. I may, as we go along, especially as I'm sharing my screen, I may turn off video just to reduce the bandwidth requirement on my network. And if for some reason I'm working from home today, so if for some reason things get disconnected, just hang in there. I'm going to hop in there from hopefully quickly soon. What I thought I will do, and one more thing, so Redcraft has been something that we have developed for probably the past decade, and it is fundamentally different in how we calculate structure from all the other existing software out there, and I hope that's going to come through. But along that line of 10 years of working, a lot of people have contributed, previous doctoral students, graduate students, some undergraduate students. But I wanted to recognize and potentially utilize two of these people that are in today's meeting. So in particular, Casey Cole is a recent graduate from my group. Right now, she holds a position at USC. Also, she works with DHEC. And so she has an extensive 2 or 3 years at least PhD work related to Redcraft. Also, Julian Raquel, I believe, is also here. And he is the current developer in Redcraft. He's actually managed to add a lot of the GUI, some of the stuff that is still under development, but some of the stuff I'm going to demonstrate today. So if you like, you're more than welcome to touch base with those two individuals. If you post questions, I'd like to see if Julian and Casey maybe can help in addressing the chats. I tend to forget to look at chat. So if you bring it to my attention, I'm happy to address questions. What I thought we're going to do today, so Redcraft in its entirety, and I will argue this as part of my presentation, is if you suspect your macromolecule of interest, in this particular case, protein, undergoes dynamics, you need to study structure and dynamic simultaneously, because if you disregard structure, you're going to get a bad, if you disregard dynamics, you're going to get a faulty structure. And starting with a faulty structure, if you're going to try to map some sort of dynamics into it, you're going to get a bad structure with a bad dynamics in order to hopefully to cancel each other. So you're going to get really erroneous results. Redcraft allows you to handle both concurrently, which means you kind of need to know how to operate Redcraft in the context of structure determination. And when signs of dynamics appear, then from there, appropriately use it for characterization of dynamical parts. So for that reason, Redcraft really has two parts to it: structure calculation and how you can refine the structure calculation part in order to resolve internal dynamics. Now, Redcraft, we previously presented Redcraft, and I'm not going to assume that all the participants in here already know how to do structure calculation by Redcraft. So what I am going to do, I'm going to present. I have a presentation, but I think we may run out of time. So I may just gloss over the presentation, just quickly give you background, jump quickly into structure calculation between now and about 12 o'clock, maybe a little bit after 12. And at that point, maybe I can then continue the discussion with dynamics. So the first half is going to be structure. The second half is going to be dynamics. Are there any questions, anything I can maybe address before we get started? In the middle, I'm hoping we can maybe, if needed, I will touch base with you to see if people want to take a break or what we want to do. If you're welcome to follow what I do as I'm going through this exercise. I'm going to try to slow down enough that you can replicate it. Now, I will recommend for you to actually, the data structure, the subdirectory that holds all the data that I am sharing with you, you might want to duplicate that into your current home directory, but maintain that original one to go back and forth, because some of the runs are already completed there. So if we run out of time, if I need to accelerate things, I can just refer to the results that are there already. So duplicate it, and we can go forward. All right, I'm going to cut out my video, and since I'm going to start sharing things. All right, I'm going to share my screen, and I am hoping you guys will be able to see. Okay, do you see my screen? That's
Yes.
okay, very good. So let me see if I can start the slide. Do you see my slideshow?
I'm seeing your instance of PowerPoint.
Okay, right. It's because it's going to somewhere else. That's not going to work. So hold on 1 second. I'm not sure which display is which one. Okay, so that's not the right one. Okay, what about now?
Perfect.
Okay, great. I am happy to share this particular presentation with you where there are two parts to it. The first part of it was geared towards structure determination. I believe this one was delivered in August of this year. Adam, correct me if I'm wrong, but I believe you can probably find the archive of the video recordings. Go back to that one if you would like to look at the structure calculation in this particular presentation before that was in August. I'm going to highlight something. So Redcraft creates the structure of a protein progressively by starting at a dipeptide. So I'm going to interchangeably, inaccurately, but interchangeably, I'm going to use in this particular presentation peptide planes with residues and amino acid residues in proteins. We know that peptide planes kind of span two amino acids, half of this, half of that, two consecutive residues. But I don't think without any loss of generality, I can make that interchange. So Redcraft starts by looking at two residues and determines the structure between the two adjacent residues. From there, it proceeds forward by appending it one residue at a time. but the simplistic representation that I have right now is very often, I would say 99% of the times, would produce incorrect structure. So Redcraft really runs in two stages. During the first stage, every two adjacent residues, so residue 1 and 2, and then 2 and 3, and then 3 and 4, and so on and so forth, is locally examined, and all the potential dihedral angles that can connect to consecutive amino acids or peptide planes is examined and ranked based on fitness to residual dipolar couplings. So in this particular instance, what I have now, let me just go back to this slide. You may start with some collection of phi size, and you have some sort of some restricted Ramachandran space, and every single one of these dots correspond to a phi psi in increments of 10 degrees that is evaluated and ranked. So at the end, you will get a list that, for example, on the top may be minus 30 degrees, minus 20 degrees, the dihedral phi psi, with the fitness to residual dipolar couplings of 0.4, and this will be ranked from the best to worst, where in this particular list, you have a minus 70, 120 with a fitness of 23 hertz. Now, there are a couple of things I want to mention at this point. Redcraft operates, examines dihedral space at 10-degree increments, as you can see here. So everything is, all phi size are in 10-degree increments. The fitness of structure to the structural models that we use assumes ideal peptide geometries. Now, these have all advantages and disadvantages, and I will focus on the disadvantages because mostly critics will think of it as that part. So keep in mind that one argument would be increments of 10 degrees may not be sufficient resolution in order to get a good structure. That's one thing that we have actually in our explorations. It seems to be sufficient, to be honest, but we do have a way of minimizing refining structure that breaks through this barrier. In addition to that, one may argue that peptide planes, the ideal peptide plane geometries may not necessarily be the case in all protein structures. So if you go on, pull out any protein structure, look at peptide geometries, very seldom they adhere perfectly to the geometry, to ideal geometries of peptides. Omega angles may not be exactly 180 degrees. They may be slightly off. Bond lengths vary. That is also another second round of error that can be mitigated by, once you have a structure that I'm going to demonstrate, you can get a fairly accurate structure with the simplifications that we are making. But it is recommended that you finally refine your structure in some sort of energy minimization in Explore NIH or Cyana, or some of the existing programs. Now, we have had quite a bit of success with Redcraft, purely with residual dipolar couplings. In fact, we have a manuscript under review with plus computational biology. It seems like it's going to be accepted. Hopefully, it will appear soon. I'm not going to expand on the aspects of structure calculation from residual dipolar couplings, other than you can calculate structure with high degree of accuracy using Redcraft and a very minimal set of residual dipolar couplings. So here are some examples. Carbonyl proton NH and carbonyl nitrogen from one alignment medium and NH from another alignment medium. We have explored a variety of combinations, anything that we can find on BMRB, as little as NH from 3 alignment media, and RDC data that can be collected from deuterated, per-deuterated proteins. So confined only to the backbone of the protein and not necessarily use just a proton in order to excite your protein. So I'm going to skip over this. And the second part is what I want to focus on today. And my focus, even though in my demonstration, I'm going to start from I'm going to start with structure. But first, I'm going to give a little bit of argument as to why we need to study structure and dynamics concurrently. So some points, relevant points, is that internal dynamics is critical for function of more and more proteins as we begin to look into it. There are a number of examples. Some are what I have shown here, and there are additional ones in reported in the literature. And this dynamics of protein is critical for their function. And these are big dynamics. We're not talking about vibration of bonds. We're not talking slight perturbation of structure. We are talking substantial structural movements are actually becoming more and more studied and becoming more critical in the function of some proteins. And I'm going to show you some examples, quick examples as we go. So here are some examples that I'm showing. NMR spectroscopy turns out to be one of the primarily main avenues of studying structure and dynamics in solution state. What I don't want to turn the focus of today's discussion is that how RDCs complement other techniques, the time span that can be studied by RDCs. Those are some of the remaining, I guess, debates in the field, whether RDCs span what kind of temporal scale. And for the most part, I think everybody agrees that RDCs fill a very needed gap in the study of the temporal scale of dynamics, but the exact details of it are a little bit disputed across different groups. So I don't want to necessarily focus on that, but residual dipolar couplings serve a particular can provide a particular approach to structure, study of structure and dynamics. And I want to make a distinction that as a final result, what we are doing, we are really coming with atomic resolution. So within under 2 angstrom resolution of not only whether there is dynamics in your protein, where it is in your protein, but also what exactly is it happening. This is like if your domain is fluctuating between 2 states, you can reconstruct the 2 states very accurately. So I want to focus on that one, on more of the computational method of reconstructing your structure. I'm going to skip over residual dipolar couplings. I think that part is there. I think it is properly discussed in the literature. So I'm going to skip over it for now, unless there are specific questions. I also want to bring some clarification and nomenclature to what we define to be dynamics. So here are 2 dimensions of dynamics. So I have structural mode of dynamics and temporal mode of dynamics. In the structural mode of dynamics, we identify and study 2 particular modes, rigid body or uncorrelated. And I have some animations to kind of highlight the differences. So in rigid body dynamics, you have domains of protein that undergo dynamics with respect to each other. However, the structural integrity of each domain remains fixed, meaning that I can talk about the domain, the structure of domain 1 and structure of domain 2, but the 2 domains undergo dynamics with respect to each other. Whereas in the uncorrelated dynamics, where essentially a portion of the protein melts away and does not maintain a coherent structure over a course of time, therefore the conventional way of discussing structure kind of fails. You do not really have a structure that you can study as a function of time. So this one becomes uncorrelated. Then in the temporal mode of dynamics is where you have either a continuous motion or discrete state jump. So for example, if we were to compare what my cursor is pointing to in that column, first column, you can see that the structures are, the dynamical parts are in continuous motion with respect to each other, whereas in discrete state, your dynamical parts is really jumping in between 2. And really the only difference between the 2 technically is that the transient states are very short-lived and the prominent states are mostly occupied. So in this particular talk, what I am going to discuss is really this particular case of discrete state dynamics. The additional ones, we discuss it in our publications, but here it's going to be too complicated to talk about, and they are a little bit on the cutting edge. Whereas this discrete state, I feel like it is at the point that we have the tools that we can kind of distribute to the community, and the community can relatively easily study proteins that undergo such a dynamics. Now, the question becomes, why do we need to study structure and dynamics together at the same time? Why is it that we cannot first determine the structure of the protein by whatever means, let's say NOEs, RDCs, everything, just structure of the protein, and then worry about the dynamics part of it later? So these are everything that I'm doing here now is all based on simulated data, so I can control the amount of error that goes into my simulations. And therefore, everything is really, from the theoretical perspective, is highly reliable. So imagine the case that you have a protein, that you have a terminal helix that undergoes two states. So at this point, I have slowed down the motion just to convey the degree of flexibility. So this particular domain of the protein is undergoing about 60-degree fluctuation from state 1 to state 2. And if you replicate that two-state jump motion and collect residual dipolar couplings and average them over the two states to come up with observable that you would get from NMR spectroscopy, and you insist on calculating a structure for that region, this terminal helix that is undergoing dynamics, its structure will be distorted. So instead of being a perfectly well-shaped helix, what you will get at the end of your structure calculation is some sort of a deformed structure, as I'm showing in green. So the effects of dynamics in structure calculation is that it will give you a distorted, sometimes severely distorted structure. Another example of it is this particular protein that's undergoing two-state jump motion, that terminal helix. If you were to impose, during your structure calculation, if you were to impose dihedral restraints to say that I know the second domain is helical, so don't let it unfold and become a distorted structure, and force helical creation, and really the only question becomes, how would that helix orient itself in the second domain with respect to the first domain? Then you will get the results that you're seeing in your right-hand side, where green and red are the two-state jump motions of the helix. And most people, when I showed this simulation, they would say clearly, you're going to find some sort of an average in between the two, meaning the orientation of the helix should be somewhere in between the two. And it turns out to be very much untrue. This helix is going to point to a completely random place because of the average and nonlinear behavior of residual dipolar couplings. The average RDCs don't particularly necessarily point to the middle between these two states. It will point to somewhere completely off. So even if you were to say, I know it's a helix, just oriented properly with respect to domain 1, it's going to be oriented funky. And if you push this, in fact, if I was to rotate the structure, you would see that this helix actually points, it's not even in the same plane as these, the green and red one. So your structure will be, the bottom line is that your structure will be distorted. Let me review our overall strategy in how we approach the study of structure and dynamics, and then I'm just going to come back, jump onto presentations. And then if time allows, I'm going to come back and continue with this presentation to show you some of the things that I'm not going to be able to demonstrate today. So the very first thing that you want to do in your study of structure of the protein, you want to be cognizant and aware at any point you may be embarking into a portion of the protein that is dynamical. So therefore, you need to have a mechanism to identify the presence of dynamics and be able to identify which part of your protein is actually dynamical. So for example, in this part, would be the entire green part. In this particular protein, it's the green and the red part. Then once you have done that, then from that point, what we do, we fractionate the protein. So we say we know, for example, dynamics starts at residue 50. So I'm going to study the structure of the protein from residue 1 to 49 and from 51 to the end of the protein. And if I manage to extract two structurally coherent, well-defined structures, and with some additional tools in comparing the order tensors that you can obtain from two different domains, you can conclude the structure of one domain is this, the structure of the other domain is that, and the two domains are undergoing dynamics, rigid body dynamics. If your structure, if your protein dynamics is actually this uncorrelated dynamics, you will very quickly see that you will fail to get structure for your protein. So therefore, it will signify that the mode of dynamics is actually uncorrelated. So once you have identified, you have rigid body dynamics, you can establish the structure of each domain separately, extract order tensors from each one, and then assemble the orientation of each one. Let me show that quickly. The identification of structure is going to manifest itself in this kind of a bump. We call this dynamic profile. I will demonstrate, I will show you my demonstrations where you see this, but during structurally rigid investigations, you will see a dynamic profile that kind of increases, plateaus at the level that corresponds to your experimental noise data acquisition level. But you will continue, and at some point you will see this major jump indicating that there's a discrepancy between a protein starting from residue 16, in this case 16, 15 onward, and the previous segments. So the two portion of proteins are operating under two tumbling anisotropy, and therefore there is potentially internal dynamics to look at. So once you have identified individual domains, then you can obtain helix, or the structure of the two domains. Then
Ome, sorry to interrupt. I'm seeing a question pop up in the chat window.
Yeah, sorry, no problem. So hold on, let me see if I can switch to my chat.
It says, what is the x-axis in this slide?
This axis, okay. The x-axis in this slide is the fitness to residual dipolar couplings reported by Redcraft. So we are observing the best structure that Redcraft can create, and what is its fitness to residual dipolar couplings.
I'm sorry, yeah, there we go, that slide.
Yeah, this is the one, right? Sorry, I think my dynamics that I have, the images that I have are big and it's kind of inundating my computer, so my presentation is not being responsive. So this is fitness to residual dipolar couplings. When your structure is void of dynamics, you will manage to fold the protein and satisfy your residual dipolar couplings to within expected error. But when I will demonstrate with Redcraft, when there are some instances that are inexplicable inconsistency between structure and data. And that is, once you eliminate the obvious culprits, then what is left over is going to be dynamics. So this could be onset of dynamics.
So maybe to follow up on Andy Luong's question, it looks like the values that you're plotting here are not on a per residue discrete basis. You've got a continuous function. What does it mean to have information here at residue 2.1?
So the plotting is incorrect. The plotting should really be a bar chart.
Okay.
But yeah, this is a bar chart that it should be at residue 1, you have something. At residue 2, you have something else. At residue 3, you have something else. So it's not really, the continuous plot is misleading, but it is per residue basis. It should be kind of treated that way. Right.
Thanks.
All right, so I'll just continue until another question pops up. So let me just quickly tell you, you can assemble the two structures. You can create the two structures. You can confirm the existence of dynamics by looking at, sorry, this is dynamics that my presentation is getting hung up on. So here are some simulated cases that we have. Two-state jump with the probabilities of 30% of the time in one state, 70% of the times in the other state. This one is a two-state jump with 25%, 50%, 25% occupancy of each one of the states. And our method is capable of reconstructing these methods of dynamics, not only giving you what the three states are, but these levels of occupancy as well. And I'm going to direct you to our manuscript that published this. These are some other examples that I have used in the past, but I'm going to disregard this for now. So when that 2 and 3 state jump, when we actually create the simulated probabilities of 0.33 and 0.67, we can calculate the back calculated occupancies is 0.35 and 0.64, very good estimates of these numbers. And the structural fitness based on residual dipolar coupling fitness, this one is meaningless at this point, but backbone RMSDs are within an angstrom, we can recreate those two. This is, by the way, the same case that if you throw the data into Explore NIH and say, I know I have a helix at the terminal helix, just do the best that you can to give me a helix. The orientation of this helix will be basically perpendicular to the plane of dynamics. Whereas our method recreates these two states with accuracy of 0.91 and occupancies of very good estimates. In the 3-state jump of 0.25, 50, 0.25, we recreate the dynamics to within 1.34 angstrom over the entire 3 states. And the gray here shows the actual, or I don't remember which one is which one. So either gray is the actual structure and red, green, blue are the reconstructed or the other way around. But you can see that they're very accurate reconstruction of the 3 states of dynamics with the estimated calculator probabilities of occupancy of fairly decent as well. So 0.38, 0.45, and 0.17. And fitness to the RDCs is actually fairly decent as well, indicating essentially what we have is a really good model of fitness. What I am going to be able to do today is to carry you all the way to the point of calculating the structures of each rigid body dynamics, but reconstructing it into these different states, how they are oriented in space, and what are the percentage of occupancies. I cannot, because that portion of the calculations takes place in a home-brewed mix of MATLAB and Maple, and we have not had the resources to kind of like compile it into one big software that does everything. I'm happy to pass along that information to anybody that needs it, our Maple and MATLAB calculations, but I'm not going to be able to. I don't have the time today. We won't have the time, and it's not the easiest thing to use those particular software platform. So if you need that part, please don't hesitate to contact me. I'm more than happy to discuss that part of it. But for today, I'm just going to stuff a little bit more tangible entity of topics of what I can cover. And I'm going to stop at this point. So once you have computed these, we're going to use the order tensor and the discrepancy between the order tensor from 2 domains in order to recreate the orientation and the occupancy of the states. I'm going to stop at this point. These are the highlights of our mathematical modeling. I'm going to stop, and I'm kind of eager to jump into the demonstrations. So I'm going to stop at this point. If there are any questions, please help ask the questions. Otherwise, I'm just going to jump straight into the demo. I feel a little bit restrained for time. Okay, so let me move things around.
Home, one question from the chat window.
Yes.
In the structure calculation, can Redcraft use NOEs along with RDCs as restraints? That comes from Aswani.
No. So again, we would love to put something like this together, but resources ran out. We are currently writing additional funds, grant applications to see if we can do this. But it is still somewhat doable if you have sufficient amount of residual dipolar couplings. The strategy that we suggest to do is to calculate a structure from Redcraft, and whatever either structural fragments that you have calculated or the entire structure that you have calculated, move that, I guess, intermediate results to explore and use something like Explore, Cyana, CNS to further refine the structure. That's the strategy that we take in my lab, but that is not necessarily the easiest thing to use. So the use of residual dipolar couplings in Explore and IH, for example, has improved over the years, but I still feel like most users of the platform have a little bit of difficulty navigating the order tensors. How do you get order tensors? How do you put things in principal alignment frame? But the general strategy is to do structure calculation in Redcraft, then go to the Explore and further refine and use additional NMR restraints there. So I hope that addresses the question.
Thank you. These are funny. If I can ask a follow-up on that. So to do the initial first part of identifying the structural regions, how many RDCs should we have? Is it enough to have, say, the NH RDCs, or we should have like multiple orientations and multiple pairs?
Right. So you always need to have residual dipolar couplings from two alignment media, because there is this concept of degeneracy that exists in a single alignment medium. So the second alignment medium is always required, but it can be minimized. So recommendation is that you need to have a second alignment medium, and the simplest thing to get would be NH dipolar couplings. So you obtain that from second alignment medium. First, then in that case, the role of first alignment medium would be to provide the most information about the structure, and then the second alignment is going to resolve any kind of orientational ambiguities. So in that case, the first alignment medium should really have, I would say, so comfortably 3 RDCs from the first alignment medium. The question becomes which residual dipolar couplings. In general, if you have NH, C alpha, H alpha, those two provide a lot of information. And if you can get the same set for the second alignment medium, you are pretty much set to study structure and dynamics from purely residual dipolar couplings. Under some circumstances, you may not have C alpha, H alpha. If your protein is deuterated, you're not going to have C alpha, H alpha. You can get the RDCs that you can obtain from a peptide plane, so carbonyl nitrogen, NH, carbonyl H, carbonyl nitrogen, carbonyl amide proton, but we would recommend to have those 3 from the first alignment medium.
Thank you so much.
For your second alignment medium, you're going to have the same set of residual dipolar couplings as the first one, then you have more flexibility in what you can use.
Thank you.
You're welcome. So keep your eyes open for some of our work. We have discussed this issue of how much RDCs that you need from how many alignment media, and in some of our previous work, and it may not appear on your radar because we publish in conferences, in computing sciences, we publish in conferences and not necessarily in journals, therefore they do not appear in PubMed. But if you do a search on Google Scholar, you will see them there, and you may want to follow them there. In fact, conference manuscripts in computer science are things that you can cite. So you can look at them, you can cite them. We also have a publication that hopefully should appear in the next month or 2 in PLOS Computational Biology. In there, we also discuss this particular issue as well. Let me move my, I'm going to share my other screen, which is my NMR box screen. And I assume, can you guys see my screen?
Yes.
Yes. Okay. So in your, I'm going to go to demo one. Okay, so let's go to the data that I have provided to you has two subdirectories. One of them is 1 Hertz. The other one is called 60 degrees. In 1 Hertz, I have simulated residual dipolar couplings for the protein 1A1Z, PDB ID 1A1Z. You can go on PDB, get the structure if you like, but I also have the protein structure here that's proteinated, that's .H. So it's proteinated protein structure that's there. We're going to use that as a template of our comparison. And we have used that structure with all the structural deformations that it has. So all the omega angles do not adhere to 180 degrees. Bond lengths do not adhere to actual, the model that we have used for our peptide geometry. So it's a real structure. From that, we have generated residual dipolar couplings, added 1 Hertz of error, and we have put it in the subdirectory 1 Hertz. We have done the same thing fictitiously. So 1A1Z does not undergo dynamics, but we have forced dynamics, a 2-state jump dynamics at a particular position. So a certain domain of the protein is undergoing dynamical changes with respect to the rest of the protein, and with as much magnitude as 60 degrees. So essentially, we have obtained one of the phi size dihedral angles of the backbone and altered it by 60 degrees between state 1 and state 2. And from there, we have generated residual dipolar couplings. So this one is a static structure. The 60 degrees is data from a dynamic structure. So what I want to do, I want to start the demonstration of structure calculation from for 1 Hertz, and then having seen what the static protein looks like, then we're going to repeat the same exercise for a protein that undergoes dynamics to see what the differences are and what the indicators are. And that's what the general plan is for today. If there are no other comments, what I'm going to do, I'm going to CD into 1 Hertz, and I'm going to show you the stuff that's there is the calculations that I have already done. So if you wanted to, you could just look at those. For now, I'm going to actually run it. So if I need to refer to these structures, I'm going to come back and go to a different subdirectory that has the duplicate of this. To run Redcraft, you say Redcraft by itself as it is. It's a command line argument that you can run, and you can do all calculations using command line. Therefore, you can run batch scripts. You can run it in non-interactive way and just run it through scripts. But if you want it to be interactive, you basically fire it up with the GUI version, and this is what you will see. And I'm going to just go through 2 examples of it, right? So quickly, the very first thing that you need to do is open the working subdirectory, which right now it's already there. And I'm gonna clear out everything, start from scratch. So I'm gonna clear all the angles that I have here. And so Redcraft, I didn't mention it operates in 2 stages. I'm gonna quickly, within the next 20 minutes, try to complete the 20 minutes to a half hour. I'm going to complete the structure calculation. I'm going to dedicate half hour of repeat for the dynamical version of the protein. So the first stage of the protein is going to give you, you're going to use the residual dipolar couplings that is in this file. And I'm not going to give you the details of what the file format looks like. You can find that somewhere else. You may want to look at the structure, the data density. This shows you I have 4 residual dipolar couplings from alignment medium 1, 4 residual dipolar couplings from alignment medium 2, and for residue 2 there is less, and Sporadically, there is less data density and more data density. This is, by the way, a very complete set of data from our residual dipolar couplings RedCraft perspective. So I understand that as a NMR spectroscopist, you may look at it and say, this is a lot of data. In my previous example, if you go look at it, or this particular example, if you want to tinker with it, you can reduce the RDCs to just backbone NH, C alpha, H alpha, and you will get structure. So this is not to convey to you, you need to have this much data. I have included this much data only to simplify things a little bit for this demonstration purposes. So the very first thing is that you select what kind of a Ramachandran space do you want to explore for your dihedral connectivity. Here I'm saying level 1. Glycines have a slightly different, significantly different Ramachandran space. You can specify the Ramachandran space, and then if you want to specify any cutoff based on residual dipolar couplings, you can specify it here. I'm going to demonstrate these two a little bit more. So you can say, I'm going to go to stage 2, and I'm going to remove everything there too. So we just have a clean slate. We have nothing else here. And go back to generating angles and say execute stage 1. And this is how fast the first stage is completed. And so if I was to go back and make some clarifications. So in this particular subdirectory, let me just create a tab. So in the same subdirectory where my analyses were completed, I can, for example, say, let's don't worry about this portion. I'm going to say plot 1.2.angles. What you are seeing is the set of all the angles that were used for residue 2. So Ramachandran level 1, it is this restrictive. So in here, I can this is more of a 3-dimensional plot where I have the Ramachandran plot, and for every one of the phi size, I have fitness to residual dipolar couplings. And you can see at this point is pretty much everybody has the same fitness of 0. So this might have been a bad example, but let's see what 3 looks like. 2 was underdetermined, 3 has more data, and therefore things are more discriminated from each other. You can see that at residue 3, the dihedral angles phi size will have fitnesses that span anywhere from 0 to 1.6 hertz. And they kind of have this kind of distribution. I don't know if this is visually compelling. To give you a better clarification, what RedCraft does produce for every one of the residues, 2, 3, 4, 5, all the way to residue 82 in this particular protein, it creates what's called an angle files. And those angle files was what I was just plotting for you. You can click on each one of them. The list will come up, and this is essentially what RedCraft has determined. The best suitable angle is dihedral 6030 for residue 2, which has a fitness of this much to residual dipolar couplings. And the worst one is this angle combination, negative 90, 120. And if I was to do the same thing for 3, you would see a similar thing, but has better results, more distributed results, because it has more RDC data. So for residue number 3, angle -50 has a best fitness to residual dipolar couplings, where if I was to do the same thing to 4, for residue number 4, angle 40-50 has the best residual dipolar coupling fitness of 0.14, and so on and so forth. This is 5th one. So between 2, 3, 4, 5, the top angles appear on the top of the list, and everything else is ranked, and the worst ones appear at the bottom of the list. let me stop at this point and see if there are any questions. There is,
May I ask a question?
yes.
So when Redcraft is trying to look for best phi, if I understood correctly, it's you asked it to choose non-glycine sort of space.
So when Redcraft is executing its work for non-glycine protein amino acids, it will use sort of like an alanine-like Ramachandran space, but for glycine residues, it will use an appropriate Ramachandran space for glycine.
So my question was,
go ahead.
For non-glycines also, like there are variations, right? If a particular residue is a pre-proline, it will have a different allowed region. So will you already consider that, or you just look at the most possible space allowed for?
So we also make a distinction between proline. We make a distinction between proline, glycine, and anything else. All right.
Okay.
So you can specify prolines, but during this first stage, you have great degree of customization. You can prime the stage not based on Ramachandran. You can prime it based on Talos dihedral restraints.
Okay.
If
Great.
you have some other way knowing that your protein is, for example, this region is helical, you can create this part fairly customizable.
Okay.
So it can be driven by other things. In here, as a simple demonstration, I'm just using Ramachandran based space.
Thank you.
Yeah. So now this brings the question of, okay, so at this point, I know what is the top candidate for residue 2 is angle 60-30, for residue 3 is angle -50-50, for angle 4 is 50-40, 5 is -60-60, and I have all this information for the entire protein. So at this point, one can conclude that we are done. All I have to do is create a structure based on this phi for residue 2, this phi for residue 3, and so on and so forth. And it turns out 99% of the time, that's going to create a really bad structure because these estimates are based on localized residual dipolar couplings, and they do not globally optimize the fitness to the structure. Therefore, you need to engage in a more global search for structure, and that's where this generate RDC or the second stage actually comes in. So during the second stage, we're going to take information from the first stage and we're going to expand and do a more exhaustive search of residual dipolar coupling structures that fit residual dipolar couplings. All right, is that point a little bit clearer? Okay, I'm going to assume silence means yes. So during the second stage, there are a variety of parameters that I am going to adjust. Let me take the time to explain that maybe, and then I can just run it. So in 2 dot angles, I had a total of, this is 182 lines. There are 182 surviving possible dihedral angles. In 3 dot angles, I have less. I have only 58 lines. This is, you can see it in the bottom of my screen here. There are 58 lines. Let's simplify this. Let's say in here, I have 200 possible phi size. Here, I have only 50. So 200 for residue 2 and 50 for residue 3. I want to create the entire exhaustive combination of everything that's in the list. So 200 by 50 is going to be 10,000. 10,000 combinations that look at now 2 fragments, a fragment that consists of residues 1 and 2. And if I was to include that exhaustive search using residue 4 now expanded to become 3 residues long, it would be exponentially growing. So at every one of them, you have 100 possible entries you want to do an exhaustive search, it would become 100 to the power of 3, 100 to the power of 4, and it will become intractable. So what Redcraft does, Redcraft starts by creating an exhaustive combination of residues. For example, I kind of closed residue 2, so residue 3 and 4. Let me start this over. So this is residue 1, this is residue 2, this is residue 3. So Redcraft will take all possible angles for residue 1 and all possible angles for residue 2. It will be over 10,000 conformations, evaluate every one of them, rank them, but allow only 100 of them, the top survivors, to survive, to be elongated by one residue, and the remainder will be basically ditched. So I'm going to execute stage 2. I'm going to use data from both alignment media. I'm going to use in stage 2, and there are some additional features which I'm going to, for now, disable. So just this, I'm going to execute stage 2. So my apologies. My explanation of structure calculation from residual dipolar couplings in Redcraft is going to have to be minimal. So I cannot give you extended discussion of what these parameters are. So I'm going to give you superficial ones. If you do not understand what I'm saying, which probably is the case, it's too condensed, please go back and either read our manuscripts or read the previous video. So at this point, at the depth search of 100, Redcraft is done calculating a structure. And you can look at what structures it has calculated by looking at, for every residue, you had an angle file. You will have an output file that gives the status of Redcraft's creations. You will create a PDB. A PDB is created that includes residues 1 through 13. And then there are 2 corresponding RedCat files that are created for detailed analysis if you want to go on the route of dynamics. So what I'm going to do, I'm going to go to residue, the end residue, look at the final structure. And you can, if your system is configured properly, if you double click on that, it's just going to bring PyMol, show you the structure. And if you want to compare it to the actual structure, you can go and compare it to this PDB that I have created there. So you can see the actual PDB is fairly globular, but the structure that I obtained was very unfolded. And if you wanted to, you can calculate the backbone RMSD between the two, and you will get some ridiculous numbers. They are 16 angstroms away. You can see that it kind of created the helical regions, but it didn't fold the protein collectively. You can start to ask the question of what is the issue in here, the issues that you may be able to resolve are, let me show you some examples. So look, you can look at an output file, for example, let's say 23.out. At 23.out, you can look, you have the combination of best phi size that have survived up to residue 23. And you have, because your depth search was 100, so far you have 100 top surviving combination of phi size that created a structure. If you look at the fitness, the progress of fitness up to residual dipolar couplings as your dipeptide plane becomes 3, becomes 4, becomes 5, and it keeps growing, is shown here. This is what we consider to be dynamic profile. You can see that the fitness to the residual dipolar couplings, it increases as expected. It plateaus based on our experimental error. But at residue 14, 15, 16, there seems to be a jump, and then another substantial jump at residue 18. So this requires an explanation. You have to look at what could be the explanations. The explanations are that 10-degree angles is not sufficient. Maybe you need to stick to 2 degrees, and it manifested itself some structural error here. So in order to resolve that, what you do, you enable this feature of refinement. You can say depth search of 100 was not adequate, in which case you will enable this feature called decimation that really is expansive in its search. I don't have time to explain what it is, but it expands the depth of search substantially. But then the remaining ones that are the issue, that could be the issue, are that there's a substantial variation, departure from ideal peptide geometries. And the only place that can come in is that this dihedral residue 2, so the Ramachandran level 1 was too constricted. What I'm going to do, I'm going to enable all the features that I have here. And let me go back to one more point. I'm not sure if this point is coming clear. If you were to look at the entire protein at the end and look at this output file, you will see that there are these points of jump. And in fact, if you go back and look at these points of jump, they correspond exactly to residues in the structure that the dihedral angles are outside of Ramachandran space and are really outliers. So the way that you can correct for those, I'm going to rerun everything. So I'm going to turn on everything here, and I'm going to clear all the previous run. I'm going to go in here, clear all the previous runs, and I'm going to basically say expand my Ramachandran space to be level 2, which is usually what we do anyways. And I'm going to execute my stage 2, my stage 1, using Ramachandran level 2, a more expansive Ramachandran space. To give you an example, for example, 3 seemed to be very restricted before. Now it's more expansive. So the 3 is including more number of phi size, and it's a more constrained, more expansive space. I can go now recreate my structure with all the exact same parameters, except I have these features enabled. I'm going to run the protein. I'm going to run the structure calculation. And now we're going to look at the progress of Redcraft. So at 4.out, things are going up, and the structure is kind of like fitness to structure is kind of plateauing at about 0.5. You can see all the dihedrals are no longer adhering to 10 degrees because of the refinement process, the further minimization that breaks through the 10-degree grid search barrier. I can now go to residue 12.out and look at it. You can see that this number is slightly inclining, but still plateaued. There is this green line that I'm not going to be able to explain what it is. You can just primarily look at this red line. Any substantial jump in the red line is indicative of clear existence of dynamics or violation of dihedrals or peptide geometry. So sometimes you may see, for example, cispro instead of a transpro. That will appear as a jump here. Then you can go back. If it is a proline that's causing this issue, you can replace it with a cispro. So in Redcraft, if you designate CPR, designates for a cispro. If it still occurs, it's some sort of an error. Let me go to 23.out, which was what I had looked at before. You can see that this behavior is changed from the previous version to this version. It is well behaved. It is staying plateaued. It basically indicates structure calculation proceeds normally. You have a good structure, And the difference between before and now was due to two constraints of Ramachandran space.
May I interrupt?
Yes.
Could you explain the numbers on the left? I see the 100 is the depth level or the search depth, but x-axis is, I think, residue, but each residue should have two angles, right? I see only
Exactly.
one.
Right, it is. So at this point, we are at residue 23. We have 23 residues. For each residue, we should have a phi psi. So this is phi psi, phi psi, phi psi.
Okay.
And that repeats itself all the way to the end, where the last column is fitness to residual dipolar couplings.
Okay. Thank you.
And the depth is no longer at 100 now. If you look, it actually exceeds. What this is due to decimation. My depth is 100. So 100 residues, 100 structures get a free pass to next round of elongation. But Redcraft also, of all the leftover structures that would have normally been discarded, will examine and cluster all the remaining structures into 2 angstrom resolution. So all structures that are within 2 angstrom bunch up together. So from those, it will select 1,000 representative structures. So each cluster may contain, for example, hundreds of structures. From each cluster, a representative goes forward that meets a certain fitness criteria to residual dipolar couplings. And that result of clustering and additional selection contributes from 101 to 249 additional, so 149 additional structures that go to the next round, therefore contributing to robustness of Redcraft. And you will see, if you were to take this data, go through it at Explore NIH and just use everything that I am using, you will not be able to get a good structure. So Redcraft in that sense is doing really well. Here at residue 51, if I was to bring up my structure, this is the structure that Redcraft has calculated so far. If I compare it to the X-ray structure and align them, I obtain a backbone RMSD of 1 angstrom. If you were to throw away some beginning end part, is even 0.8. So at this point, Redcraft has managed to create a structure that is fairly comparable to the X-ray structure. If I was to study the status of how successful is my structure calculation, again, I go back to .out file and I look at my dynamic profile here. And this dynamic profile, things are a little bit slow here. So the dynamic profile indicates no anomalous behavior. There's an increase in behavior, gradual incline, but still plateauing at approximately expected experimental error is this much. Green means basically all structures that have residual, that have RDC fitness above this 0.5 to within 0.85, they were considered, they were clustered into 2 angstrom bunch, and representatives were then included as part of calculations for the next round. So green basically tells the ones that additional structures that basically were allowed to go in addition to the 100 exempted structures, all these structures in this range were allowed to go to the next round. I'm gonna, at this point, things are getting a little bit slower, maybe because too many of us are using the same thing. So .out file is good at 58, the PDB file. I'm gonna compare it with X-ray structure again. So we are still at 1 angstrom, and the structure is proceeding forward, and so far it has gone this far. And as you begin to look at the structure as it grows, it goes into this helix, turns, and comes out. If you like, we can go back. Let me, while that is running, I'm going to get out of here and go to the example that I have already run. And what I can do, let me so the structure that it has, if you look at the .out file, the dynamic profile file indicates no problems. And if you align the two structures at the end, you will get this. You are at, again, 1.1 angstrom with respect to the X-ray structure. And the whole run on NMR box takes less than half an hour. Right now it's taking more because the system is probably overloaded, but generates a fairly successful structure. But again, to stress, I had to correct for Ramachandran space. I had to correct for refinement, and I also corrected for shallow searches. It's more extensive search. Let me stop at this point, see if you have questions. If not, I'm going to repeat this exact same exercise for the protein where there is actually dynamics in it. And I'm going to just terminate this prematurely. I don't see any point to it. So are there any questions at this point? Right, I'm going to stop my run. And what I'm going to do, I'm going to basically change my workspace and go to 60 degrees. In the 60 degrees, there was. So let me clear all the angles. Again, so if you look, we are only using the same exact amount of data. There's nothing different. And I'm going to keep everything the same as before. And I'm going to clear all the previous runs just to have a fresh start. Now, if I was to run this, what is going to happen is I'll run this. So for first stage, I'm going to execute. Somewhere in this protein, there is dynamics. And the question is, can we identify what it is? And can we retrieve the structure of each domain? So I ran stage 1 with Ramachandran stage of 2 for non-glycines. And I just said for the same thing for glycine, I'm not going to do any cutoffs. In the interest of comparing apples to apples, I'm not going to make any modifications to this. This is all the same thing, and I'm just going to run it. I think this number is different because only I'm trying to run it faster. But otherwise, if you were to include the same numbers from the previous run, which I believe it was maybe 1 or 1.1 here, it would give you very similar results. It just would be slower. So if I was to start this, so there is going to be a bit of I'm just going to be quiet at this point. I need this run to be completed in order for me to actually show you the results. Maybe what I can do during that time, if there are any questions, I realize that I stuffed a lot into structure calculation part of it, including what these parameters are, including maybe depth search. So if you have any questions regarding that, we have about maybe 10 minutes before RedCraft gets to a place that I like to discuss. If not, I'm just going to monitor the progress of Redcraft. So it gets to residue 15. I'm going to look at 15 dot out. It seems to be well behaved. 16 dot out. The same thing is well behaved. I'm going to look at the structure. It looks, it's a single helix at this point. It looks good. So at 21 dot out, everything's still good. Maybe this is worth comparing to the actual structure. And so we're at about 0.76 angstrom, about 0.8 angstroms so far. And I'm not sure if I mentioned that there is about 1 hertz of error randomly incorporated into residual dipolar coupling. So these are not perfect RDCs. At 27, it's still well behaved. There is no anomalies here. To be honest, I'm not exactly sure where the point of dynamics is. So we kind of have to wait and let it run. It looks good structure. I'm not going to worry about obtaining the backbone RMSD. Is going to be good. 29. It looks good.
May I ask a doubt at this stage?
I'm sorry?
Could I ask a question?
Yeah, absolutely. Go ahead, please.
So, so if I understood correctly, we're going to get a hint about the dynamics by the RDCs that we measured, right? And as we are using those RDCs to calculate the structure.
That's correct.
And we will get that in the dynamic profile, and we'll see a jump. And we make sure that the jump is not because of like bad sampling of the 5 size space, and then we figure out what the dynamics are.
Right. So let me tell you the reasons why you may see a jump. Okay. So you may see a jump because you will not, because your angular constraints is too much. So either we have never seen this kind of a jump because of adhering to 10 degrees. And if you are interested, in fact, you can either conduct that experiment or I can conduct it here by, if your Ramachandran space is allowing, it includes the right angle. At 10-degree increments, you will get a good structure, reasonably good structure, even though that may be counterintuitive. You will get a good structure. So if your Ramachandran space, if your proper angle, the actual native structure has dihedral angles, that's really far outside of your allowed Ramachandran space is one reason that you'll see a jump. And the reason we use 1A, 1Z as our example is because it has numerous one of these. You may see another jump if you have a cis pro. But cis pro actually has a very distinctive blip, a jump. I'm not going to say too much more other than cis pro. You will also see this kind of a jump if you have misassigned your data. So we have used this process in the past to assign, to provide assistance to assignment of data when chemical shift data may be ambiguous. So at some point, you may have 2 possible sets of RDCs that fit next. You can put one combination, run your program, and see if you run through your structure, everything is good, then that's the proper assignment, that's the proper structure. However, if you see a blip here at that particular location, then you can swap the other potential candidate for assignment, run, and one should fit, the other one should not. So a misassignment is also another potential candidate, but in the absence of those, so if you're running at Ramachandran level 2, if you are minimizing your structure properly, you are not going to see that blimp unless it is dynamics. And I can show you how you will confirm further that there is dynamics. So there is more redundancies built in here. Does that kind of maybe?
Yeah, I think, yeah. I was wondering why there's a generate RDC tab on the Redcraft GUI.
Ask that question again. Why do we what?
So in the GUI, generate angles is the first tab, and next to it you see generate RDC on the GUI.
Yeah. So which part of it you're talking about? The
yeah, there.
Yeah.
So you have a generate RDC tab.
Oh, yeah, no, sorry. This was an error that we corrected. So this is not the latest version of Redcraft. We didn't have time to update everything here. So if you download Redcraft, the latest version, this says generate structure. It's not generate RDCs.
Okay.
It's a typo.
Okay.
Yeah, sorry. So we are now at residue 44. Is calculating 44, but it's done with 43. 43's dynamic profile is fairly stable, is doing well. Let me see, where did it go? So I need this to get to the place that the dynamics actually starts. So it wouldn't be probably until, I don't know, middle point to end of the protein. So I have another 5 minutes or so, I suspect, before the dynamic portion kind of presents itself. Let me at this point address a different thing. So one of the redundancies that's built, once you see a blip in this dynamic profile, that indicates that you have a dynamical region. But in order to establish, are you dynamical and how much dynamics, you need to get order tensors. And the way we get order tensors, at least in my lab, we use a program called RedCat. RedCat, we have tried to incorporate it here so you can stay in one integrated environment and work. But this one is still in its nascency, and Julian is developing this further. But for now, I'm going to run RedCat. I'm going to run RedCat by itself. So you can run RedCat, and here you can just directly bring in RedCat files from whatever Redcraft is producing. So for example, I'm going to go to, this goes, let me go to 60 degrees. And in here, we went as far as, let's say, I don't know, let's say 4. So I have alignment medium 1, alignment medium 2 for 44. I'm going to load alignment medium 1. Data gets loaded. Whatever is tick marked on the side are things that have RDCs and can be analyzed. Whatever does not have RDCs is indicated by 999, therefore excluded out of your studies. If you're not familiar with this program, we have documentation you can follow. The simplest thing is to just say, go to solutions and say generate report. Here you indicate a threshold, what RDC errors above which you should just exclude because they're outliers. So as it is, I'm just going to use that. This will run. It will generate a variety of information that's useful. Of particular interest is this, which is the ordered tensor, and is this, which is an ordered tensor decomposed into order parameters, and this information, which is dA and R, which is used in order to quantify essentially the strength of your alignment. So dA is a function of what nuclei you're using. So whether you're using carbonyl nitrogen or carbon nitrogen, or whether you're using NH and so on and so forth. Usually NH is the one that's used. So this dA and R indicate they're derived from these order parameters and indicate the attributes of your alignment. The higher dA number indicates the strength of alignment, and R indicates how axially symmetric is your alignment. Now, if you obtain this information from two disjointed parts of your protein, and they are approximately the same thing, then your two domains are static with respect to each other. However, if these numbers compared between two domains are different from each other, then the two domains undergo dynamics with respect to each other, which is what I am hoping to establish here. So I'm gonna stop this at this point. I'm gonna put this away. Let me monitor to see what's happening. So at residue 56-ish, so everything's still behaving, so it must be coming up soon. This protein is not that big. It's only 70 residues long, or 80, sorry. So bear with me. Let me tell you a couple more features here. What is kind of useful about residual dipolar couplings as opposed to NOEs for structure calculation, because all the data comes from local residue. In NOE, your residue number 50 may have an NOE connection or distance constraints relative to residue 1. So residue number 1 and residue number 50 need to be actually looked at in the same together because they are essentially connected to each other. Whereas in residual dipolar couplings, that's not the case. In Redcraft, I could have easily said, for example, give me 2 instead of 283, let's say, for example, 50. I could just study this segment of the protein. I could have said, give me structure of residue 25 to 50 by itself. And I could have just isolated and focused my study calculation for this region. And this is something that we're going to use going forward. So there was a chat, sorry, I was looking at. Now, at some point when there is an onset of dynamics, let me see what's happening here. So this is potentially it. You can see that your dynamic profile, this particular figure, we call it dynamic profile. You can see that starting at residue 60 is beginning to incline. So at this point, we have resolved our Ramachandran space is large enough. You can go and if you like, you can run it with even more expanded Ramachandran space. But at that point, if you were to submit that structure to PDB, PDB would have a red flag saying that you have Ramachandran inconsistency. And as long as you know your assignments are correct, as long as you know everything else is in place, all the, the simpler culprits, then this is potentially the point onset of dynamics. And I'm going to let it go a few more residues, but in the interest of time, I may just terminate it. So we are at residue 63 is out. And you can see that it is beginning to go up, and it's kind of like, it's really not coming back. It was not just an anomalous one residue blip. If that one residue blip was a proline, this could have been, and the blip that you saw went up, came back down, it could have been interpreted as a cis proline. But at this point, it is not, and I'm gonna let this run a little bit longer, just maybe up to residue 70 or so, just for it to be clear that this is not returning back. So you can see that it is still up there. Now the question is, from this perspective, there is an issue there. And since we have resolved all the other problems, the only explanation that remains is that that point is an onset of dynamics. The question is, what is it doing to our structure? Let's look at the structural comparison. And you can see that at this point, this is the point that apparently appears to be onset of our dynamics. The structure, instead of turning and becoming a helix, is going in a random arbitrary direction unstructured. So this is again another demonstration of how residual dipolar couplings reported from dynamical region, if it is forced to produce a structure, it's going to give you, erroneous structure. So I'm going to stop at this point. I think the point is clear, unless somebody wants to challenge me on that starting residue 60, there is some sort of an anomaly. And if you let this go from residue 60 all the way to 83, that peak does not return back. And you can check that in your original data set. What does that mean? So there is anomalous behavior at residue 60. Now, I'm going to terminate this for now. So bear with me. I'm going to just set the stage here for the next round. What am I going to do is that I'm going to copy my 60 degrees into, I'll tell you why. All right, so up to residue 59, we seem to be okay because the dynamic profile indicated everything is okay. So I am going to accept 59.pdb as a legitimate structure up to that residue. At residue 60, there is an anomalous behavior taking place. So what I am going to do, I'm going to accept residue 1 through 59, and I am going to investigate residue 60 onward as the potential domain 2. So what I'm going to do, I'm going to change my workspace. I'm going to go to domain 2. In domain 2, I'm going to open it. And everything, you can run stage 1 again if you like, but that's already done. What I'm going to do, I'm going to clear all my output from stage 2. And I'm going to run this from residue 62 onward to residue 80, 83, and I'm going to keep all the run parameters the same thing. So I'm going to now execute this stage. And you will see that I'm going to begin to have, I started at residue 62 to just give it 2 residues of buffer region. So you can see then if I was to look at the output at, let's say, I don't know, residue 68 right now, you can see that. So residue 68 has the same kind of a behavior that it goes, increases, plateaus, it's reasonably well behaved. If I keep looking at the rest of it, so I'm getting at the end of the protein 77. Hold on, let's look at 77 that out. You can see, so it actually went up again. Is that true?
The point of dynamics may be at residue 69, I think, for this one.
No, I mean, so there was that jump at residue 60. So maybe it was at 69. Maybe I prematurely terminated it, but it may be at residue 69. So I can look, so I can examine that. Is it 69? So up to residue, it's a little bit too small. So up to residue 68, 69, it seems to be behaving, is jumping back up again. So let's say 72. Yeah, so at this point, it's going back up again. So the point of dynamics might have been, there are actually 2 points of anomalies, so residue 60 and residue 69. Once I isolate each one of them separately and study each one of them separately, I should get a more accurate structure. But I am almost sure that I had done this for, let me refer back to the original data. Adam, where's the data should be, the original data should be collected. Where can I find the data?
Let me take a quick look.
It was in public?
It's in public events, I believe.
Events, yeah. It was
All capitals, yeah. And then there's a subfolder for you.
Okay, yeah. Let's just say, I'm going to copy this to my home subdirectory and I'm going to change my focus to that one. I'm not sure why exactly this is happening. It may be because I need to repeat stage 1. Let me see how it would look like if it had gone all the way through, which is what I have here. So it does seem like there are 2 points of dynamics. So this is a really complicated case. There is one case at residue 60, one residue at 1 point of dynamics, at potentially residue 69 or 70. It's hard to make the numbers. So let's look at residue 60 through 70 in our run. So in our example that we ran here, it was dynamics. No, not that one. So I can obtain, so this is the one, if I was to look at this and look at the structure at residue 70, no, that's not the one. Oh, it was under domain 2, sorry. So the same region of protein that it was unstructured, now that I allow it to be studied from residue 60 to 70, before it was unstructured, now it's a perfectly well-formed helix, and it is very, it's a very good fit. Now we're kind of coming to an end. Let me just quickly tell you what would be the next step of confirmation would be. So at this point, you run fragmented structure calculation. So you know some sort of an anomaly occurred at residue 60. You obtain a fragment from residue 1 to 59. Then you obtain another fragment from 60 to 70. And now you want to explore the hypothesis that you're undergoing dynamics. And at that point, you will run red cat. So let me go Redcraft Dynamics. I'm going to do this quickly for each one of them. So 59 was as far as I went for residue number 1. I'm going to generate the report for it. And again, I'll cut it out 1 hertz. This is the order tensor that I get for alignment medium 1. I'm going to close this file. And I'm going to go and load the file that I get from my second domain, residue, let's say, let's say 70 or maybe 69, just to be so. And then if I generate the report again here, I will have both results kind of appended here, where my first alignment medium, my first domain had you can either compare your order parameters specifically or DAR values. So let's go DA of value of -9.4 is for domain 1. Domain 2 is going to have a DA value of -10.27. This is somewhat substantial. But if you look at R, has a value of 0.027, and for R here has a value of 0.18. These collectively constitute existence of dynamics. So at this point, you can conclude that that middle residue that caused some sort of a jump, let's say residue 6970, the bump is not because it's a cispro. The bump is not because it is Ramachandran spaces too constrained. The variation in the order parameters itself will indicate that there is clearly dynamics between the 2 domains. What you do have is structure for domain 1, structure of domain 2, both reliable. You have a confirmation that there exists dynamics between the 2 domains. And from here onward, you can use the discrepancies or the differences between observed order tensors from domain 1 and domain 2 in order to reconstruct the atomic position of the domains with respect to each other if it is undergoing a 2 or 3 state dynamics. And that work has been published. So I know I'm running out of time. The work has been published. If you want to look at it, what I can do, maybe there is, if you look at my list of publications, it is actually published in, Casey, are you here?
Yes, I'm here.
Okay, do you remember what the name of the title was or where it was published?
JTCT, Journal of Theoretical Chemistry and Computation, I think.
Yeah, it was.
I can send the link in the chat.
Okay, sure. Why don't you do that while I'm closing? So you can go and look at, if you look at our publications in Redcraft and Structure and Dynamics, it should be clear which one it is. And that one highlights the process that I showed today, but it goes farther into how you can get order tensors and do further analysis in either MATLAB or Maple in order to reconstruct the orientation of your domain 2 that's undergoing the fluctuations and reconstructed, oriented in conformation 1 and conformation 2. It will recreate both conformations for you. It will also give you the estimated occupancies of each state. So whether it was 50-50, whether it was 30-70, the limitations of the method is also described there. You cannot go beyond occupancies less than approximately 20%. So if something is 10% occupancy and 90% occupancy of the second conformation, you cannot make that much. It's usually 20% and above. And it also depends on noise level, how much noise you have in your data. Clearly, the more noisy data, the less you can do with it. So I'm going to stop at this point. I hope this was somewhat useful, but what you can do, you can just download Redcraft, download this data, maybe get this video, and you can just follow it in order to reproduce what I have done in order to establish how it can be used. Please feel free to contact me or anybody in the lab if you have any questions. Redcraft is on GitHub. You can just get it from there, or Bitbucket, I'm sorry. Or you can get it from our website. So if there are any questions, I'm happy to answer. Otherwise,
Homay, this has been fantastic. And I do have a question. Tell me if this is a stupid question or not, but it strikes me that this might be something, not all IDPs and IDRs are created equal. Is this an approach that could be used to look at the relative extent of dynamics in IDPs and IDRs?
I believe so. It's something that, yes, in my opinion, yes, absolutely. If you were to broaden the other people's inputs, people tend to challenge what RDCs can do. But please keep in mind, if you take residual dipolar couplings and stick it in Explore and IH, you're going to get some really bad, nasty structures.
Oh man, you're not kidding.
Right. So, and it's going to be inexplicable. You're going to have things that should look like a helix, but the structure that you get out is basically going to look like, unstructured regions. So yes, the Redcraft is designed for that. And depending on how you want to use it, you can explore the hypothesis of, is there dynamics? You can from there reconstruct the structure of each domain of dynamics if it exists. And from there, you can assemble the structure together. So this is all theoretically very sound. And in practice, we have demonstrated it a number of times. The only problem is that for us to demonstrate it, we use synthetic data. So if anybody has got actual data, experimental data, we are happy to analyze it for you. We are happy to tell you how to analyze it. We can work with you to explore the possibilities of dynamics.
Homay, is the synthetic data something that's coming through MATLAB scripts?
Synthetic is coming through RedCat.
Okay.
So we use RedCat, Redcraft combination to generate, reconstruct. So we start from a PDB, use RedCat to generate RDCs. We take the RDCs, put them in Redcraft to get structure and dynamics. MATLAB and Maple come in when we have confirmed dynamics. We get the order tensors, and in order to decipher what kind of a dynamics would map one order tensor to another order tensor, that is done in Maple and MATLAB. It's mostly optimization.
Okay. As you may know, we actually have MATLAB installed on NMR Box now, open to the community. So if you've got some utilities that you want to just drop right in, if that would help, we can host those.
It's right now it's mixed between MATLAB and Maple. So our plan is, yes, absolutely. If I can consolidate that to something that users can just come and give the information that they have without much tinkering with it, yes, absolutely. I may just put it up there.
Okay, great.
If I can ask one last question. So from the analysis, we get populations, but do we also get a hint of the time scale of the dynamics?
No. The time scale of dynamics you have to get by some other means. So either it has to come through your experimentation. What I have proposed to do is, it's awaiting for somebody to do this, is, so there are two software out there that very well work with the final results that we have. So at the end of this, you have the structures determined, the two states determined. The question is, how does it go from state 1 to state 2, and what's the time scale of that transition? So you can either use NAMD that has targeted NAMD, which is a molecular dynamic simulation, but it has a very specific tool called targeted molecular dynamic simulation. This is where you say, you give it two structures and you ask NAMD to morph one into the other one by keeping adhering to principles of biophysics. The other one is GROMACS. GROMACS also has ensemble, what is it called? Structural ensemble, that you can give it an ensemble of structures and it will explore transitions in between those two. So those may give you time scales if you want to go computational. But at this point, I would say your estimate of a time scale still is your specifics of your data acquisition.
Hey, there's still an incentive for doing the experiment, relaxation dispersion.
Yeah, exactly. So that part of it comes from the atomic resolution reconstruction of the structure comes from RDCs, but all the information that fills in what is the time scale, potentially even the magnitude of dynamics to confirm the RDC reconstructed structures comes from relaxation information.
But for the determination of the structures itself, if I understand correctly, we don't need the timescale information to start with.
That is correct. For dynamics, you don't need to have the timescale. You just know that there's dynamics is going from here to there, but you don't know in what timescale.
Yeah, thank you so much. It was great, great
No problem.
presentation.
My pleasure. So everyone, I mean, I don't know how this comes to an end, but if anybody wants more additional information, I'm happy to provide more of this. If you want to expand on this, if you want to continue this, even if there's only one or two of you want to see this, it doesn't have to be part of NMR Box formal seminar, but we can hold it as part of NMR Box, maybe more private discussion.
Hey, I also want to extend my thanks to Julian and to Casey for their contributions, and thanks to everybody for showing up. Andy, nice to see you, or at least your avatar here. And Homei, this is, every time, and I was struck by your last workshop, but this one as well. This is a resource that has been underutilized. You've made a tremendous contribution to the field, and my hope is that we can help with its penetration as a result of getting it promulgated on the NMR platform. It's absolutely a fantastic set of tools you've developed.
Thank you. And NMR Box is going to be indispensable in trying to get this to the hands of users. As developers, we develop things assuming users they just know what to do. But this removes a whole lot of complexity for users. And in fact, NMR Box for people, the same program that runs on my desktop, it's a beefy desktop, runs an hour and a half, 2 hours. Our NMR Box servers runs in about 10 minutes. So there are a variety of reasons why to use NMR Box. It's not just that everything is there and ready to go. It's a beefy hardware too.
Thanks for the plug, Homei. Yeah, we've got roughly 40 cores on every single one of the VMs, and users are entitled to run 3 simultaneous sessions. So we've got some computing power available to you when you need it.
This is not easy to obtain. So thank you very much, everyone.
Okay, and final tip of the hat to Adam for being the major domo of all of our workshops on the NMR Box platform. It's no small feat. There's a lot of stuff that goes on behind the scenes that you guys should know that Adam is a bit of a magician at making the difficult look simple.
Yes.
That's very kind of you, Jeff. But let's put the spotlight back on our featured guests. We've got some really fantastic presentation materials that come through.
Yeah.
All right, then I'll close out the session here. The video feed and slides, if you want to share them, Homei, I'll post those all on our website on the page that everyone already has a link to. And as we fill out our year-long monthly seminar series, we'll post that on Slack. We'll post it on the website. So stay tuned, and we hope to see you at a future seminar.
Very good. Thank you, everyone. Bye.